welcome to your exam 2 review. And let's jump right in. All right, so first touching on some basics, kind of reviewing a little bit. So aqueous chemistry, why do we need this? We need to know water is a polar molecule. This should be pretty familiar to all of you. It has a tetrahedral geometry, so in its three-dimensional shape. Right here, it doesn't really look like tetrahedral, but in its three-dimensional shape, with its lone electron pair, it is tetrahedral. Oxygen carries a partially negative charge, and hydrogen carries a partially positive charge. This is important because we know water can form hydrogen bonds with a variety of substances and interact with other polar substances. This is important when we talk about wanting to dissolve other substances in water. For example, hydrophilic nonpolar molecules, when they interact with water, they are hydrophobic, which means water fearing. And usually it forms a layer of constrained water molecules around those nonpolar molecules. We also have hydrophilic molecules, which are water-loving, as the name says. Those are usually polar molecules because they can form hydrogen bonds and other ionic interactions with water. And lastly, we have amphiphilic molecules. Those contain both hydrophilic and hydrophobic portions. So they have both properties and interact with water differently depending on the size of each portion. This leads us to the hydrophobic effect, known how nonpolar molecules interact with each other in water. So if we just have lone nonpolar molecules in water, they get hydrated, as we saw in this picture right here. So we have that layer of water molecules forming around that nonpolar molecule, which leads to a loss of entropy. So this is not very desirable. So usually those nonpolar molecules aggregate together in water. For example, if you think about putting water in oil, usually it forms a layer on top of the water because it doesn't want to interact with the water. However, if you stir it, you see like little balls of oil forming in water as the oil molecules aggregate together. This is favorable because we have higher entropy that way, so more chaos and lower energy. So the nonpolar molecules will arrange in a way that the nonpolar ends, for example, are avoiding contact with water. So this would be, for example, a protein that has both hydrophilic and hydrophobic regions, but it would ball up in a way that the hydrophobic regions are away from the aqueous solution, and the hydrophilic regions are on the outside. And same thing just for nonpolar molecules. They aggregate in a way to avoid large contact with water. Talking some more about the amphiphilic molecules. So again, they have a polar and nonpolar portion. If you think about, for example, a bilayer or lipids in our membranes, this should look pretty familiar. We have that polar head group and a nonpolar tail. If they form a micelle, it will look like this. So we will form a hydrophobic core with all the nonpolar tails pointing inward away from the aqueous solution, and the polar head group on the outside interacting with the water. Instead of a micellular structure, we can also form a bilayer, which would be a two-layered sheet that also has a hydrophobic inside and the polar head groups on the outside interacting with water. Both of these structures are thermodynamically favorable because they avoid contact with water. A bilayer also serves as a diffusion barrier, so it prevents molecules from diffusing from one end to the other of this bilayer, which is important when we talk about cell barriers and so forth. All right, another important basic is acid-base chemistry. So the ionization of different molecules, so adding or removing protons from a molecule in order to ionize it. So when we talk about water and water being ionized, we produce a hydronium ions and hydroxyl ions. So usually we only talk about H plus protons, but we usually don't just have protons floating around by themselves. Instead, we have that H3O plus formation. So a water gains a proton and is ionized from H2O to H3O plus and OH minus. So this would be 2H2O to make that equation correct. Next, we have the dissociation constant K, which talks about how likely a substance is to dissociate into its ionizable parts. For water, for example, that would be the Kw water constant, which is essentially the concentration of protons times the concentration of OH minus over the concentration of water at 25 degrees Celsius. That would be 10 to the negative 14, which would be a neutral pH. And the concentration of H plus and OH minus are inversely related, as you can see right here in this graph, which makes sense. I already mentioned pH in the last slide. It is a logarithmic scale and talks about the concentration of protons. So 1 unit, this is important to remember, 1 unit is equivalent to a 10-fold difference. So a change of 1 in pH actually is a change of a 10-fold magnitude with the concentration of protons. The scale is also inversely related to the concentration. So the more basic or the higher the pH, that means the lower the concentration of protons, and the other way around. The more acidic, the lower the pH, the higher the concentration of protons. Another important thing is acids and bases. Acids can donate a proton and usually form those H3O plus ions. Bases can accept a proton from the H3O plus and therefore take away the positive charge. And also strong acids and bases usually completely dissociate into their ionic forms. So you will see only the ionic form once they come in contact with an aqueous solution. Weak acids and bases only partially dissociate and they will form an equilibrium. So this is what I'm talking about right here. This would be a weak acid. It would only partially dissociate and form a conjugate base and its conjugate acid. This brings us to the tendency to ionize, the pK, which is related to the dissociation constant K. So again, K is calculated by the concentration of each ion to the concentration of the undissociated form and water. In this equation right here, imagine that there is a water molecule. And the pKa is the log of the dissociation constant and is a measure of the strength of an acid. For this, you need to know that for weak acids, the pKa is high, and for strong acids, the pK is low. Also important is to understand the relationship between weak acids and conjugate base. So this here is a weak acid. It has a conjugate base right here. Like I said, imagine that there is an H2O molecule which would be a weak base, and has a conjugate acid, which would be this right here. This is important when we talk about the relationship between pH and pKa. So we have the Henderson-Hasselbalch equation, which relates those two to each other. So the pH of a solution can be calculated by the pK, so the dissociation constant, the log of the dissociation constant, added with the log of the conjugate base acid pair. And if the pH equals the pK value, then the concentration of conjugate base and conjugate acid is equal to each other. Another important takeaway from this is that if the pH of a solution is larger than the pK value of a substance, the substance will be fully deprotonated. It will have donated all of its protons into the solution. If the pH is lower than the pK, the substance will be fully protonated and carry all of its protons. All right, in living systems, we want to prevent large pH changes. We need to be able to regulate our pH because proteins can denature in certain pHs, which can irreversibly cause damage. And too large changes in pH can cause acidosis or alkalosis, which both are not very desirable. So we need to be able to maintain a relatively consistent concentration of protons in our bodies. This is achieved with a weak acid conjugate base pair system that will usually try to maintain equilibrium. So for example, we have the carbon dioxide and water reaction right here, which is due to normal respiratory function in our bodies. So we produce carbon dioxide, which will react with water to form carbonic acid. And then that carbonic acid will dissociate into bicarbonate and hydrogen ions. We want to maintain a relatively stable concentration of hydrogen ions. So even though we produce more CO2, we want to make sure we don't produce an excess amount of H+. So we need this buffer system, which will reversely or in reverse keep an equilibrium between CO2 and H+. So many other physiological processes also produce protons, and we need to be able to remove or catch those excess protons to prevent acidosis. So for example, we can encounter metabolic acidosis. Again, byproduct of lots of metabolic functions is excess acid. If we produce too much, we will encounter metabolic acidosis. Respiratory acidosis, I also just mentioned this, can be due to impaired lung function, where we cannot get rid of excess CO2 and produce too much protons. We also can have or encounter metabolic alkalosis, which is a rarer event and would be caused by excess H+ secretion. So we're getting rid of too many protons and have a too basic pH. And lastly, we can encounter respiratory alkalosis, which, for example, happens when we hyperventilate, when we get super anxious or stressed, where we exhale very rapidly and we release excess CO2, and therefore have a too high pH in our system. So overall, we want to make sure that we maintain a physiological pH of around 6.9 to 7.4, which is why we need buffer systems inside of our bodies. All right, jumping into the next chapter, protein structure. This should be pretty familiar. If we think about the last exam, so we know that amino acids are building blocks of proteins. We have that carboxy group right here, and the amino group right here, the alpha carbon, and then the R group, which is important in determining properties of an amino acid. Different amino acids are linked via peptide bonds. So we have the N-terminus right here, and that is linked to the C-terminus of another amino acid to form a peptide bond. There are 20 different amino acids, and they can be grouped into nonpolar / hydrophobic, into polar and charged amino acid. Another important property of amino acids is that they are zwitterions, so they carry a negative and a positive charge, and they can be protonized and deprotonized. Remember the relationship between pH and pK here. So NH3 has a pK of 10, and COO, the carboxy end, has a pK of 4. So depending on the pH, these will be deprotonated or protonated. And lastly, another important structural feature of amino acids is that they exhibit a handedness. So they are asymmetric, or also called chiral. So the alpha carbon can be arranged with its side chains, either in a L manner or a D manner. So depending on which way those side chains are pointed, they exhibit a different handedness. The only thing that you guys need to know about this is that amino acids in proteins or in our bodies usually exhibit the L form. Okay, and then grouping the amino acids into different groups. So there are non-polar amino acids. Those have side chains that only weakly interact with water, and they don't often participate in chemical reactions. So some of these can have aromatic side chains or just regularly uncharged methyl groups, for example. And these are often found on the inside of large proteins, as we already talked about. Next, we have polar amino acids. Those can strongly interact with water because remember, polar interacts with polar. We have, for example, cysteine here, which has an important property. It can form disulfide bonds through oxidation. So two different cysteines can form a disulfide bond between their side chains. Histidine, for example, carries this imidazole ring, which can act as a good buffer system. It can ionize at physiological pH and therefore act as a good buffer. Most of the other groups usually carry an OH group or an amino group that can be polarized. And then lastly, we have charged amino acids that always carry a charge and are usually found on the exterior of molecules and proteins, and they participate in chemical reactions. So we have, for example, negatively charged amino acids as aspartate and glutamate. Those carry a negative charge right here. And then we have two positively charged amino acids, lysine and arginine, which carry a positive charge right here. All right. Next, we know that amino acids are linked via peptide bonds. So within a peptide, amino acids are referred to as residues because they form those peptide bonds and they only have their residual atoms essentially, since the other two contribute to the peptide bond. So we have a hydrolysis reaction right here. So the carboxyl group reacts with the amino group. We release water and form this peptide bond right here. This creates that C and C backbone, which is the important backbone in protein structures. And the electrostatic properties of a peptide depend on these side chains. As for naming of different peptide chains, if we have two amino acids, it is called a dipeptide. So we have two. It's three amino acids, it's a 3-peptide. An oligopeptide if it has more than 15 side chains. A polypeptide if it has more than 100 amino acids, and then a protein if it has more than 150 side chains. If we name a peptide, we can either use its full name, which would lead to a really, really, really long name. So we usually use the 3-letter code, or if it's a really long peptide, we use its 1-letter code, and we always start at the N terminus. When it comes to breaking down proteins, there are exopeptidases, which can act from the ends of the chain. So from the N terminus and C terminus, they can act and break down the peptide bonds, or we have endopeptidases, which can act from the middle of the chain. All right. Another important thing to know for your exam is how to determine ionization states of amino acids and proteins. So remember the pK value, the tendency to ionize at a given pH. So depending on what the pH value is in a solution, the amino acid will be deprotonated or protonated, and that depends on the pK value of the amino acid. You probably do not have to remember this, but know roughly where these are located. So know how to look up the pK value of different amino acids and what they mean. And once you know how to determine the ionization state of an amino acid, you can also determine the overall charge of a protein at a given pH. Related to this is the isoelectric point, the pH at which an amino acid, protein, peptide carries a net charge of 0. So this depends on the ionizable groups on an amino acid, and is calculated by adding up the pK values and taking the average of those two values. This is fairly straightforward if I have one amino acid that only has 2 pK values. However, if my amino acid has more than 2 pK values, or I have a protein that I want to determine the isoelectric point for, I need to know how to calculate this. So what I do is I take the pK values that surround the 0 net charge. Obviously, this depends on a given pH value, so make sure you take that into account. You figure out what the 0 net charge would be, which pK values surround that 0 net charge. You add those two pK values together and divide it by 2, and that will give you the isoelectric point. For asparagine and glutamine, which are charged amino acids, you take the average of the 2 lower values, and for lysine, histidine, and arginine, which are positively charged, you take the average of the 2 higher values. But overall, know how to calculate the isoelectric point and how to determine ionization states of proteins. I'm sure this will be pretty important for your exam. All right, diving into the next chapter, protein structures. So if you remember what the peptide bond looks like, let's jump back to the slide right here. We have this bond right here, this double bond, and due to this double bond, the electrons in the peptide bond actually exhibit somewhat of a delocalized nature. So the electrons from here kind of move around between these two bonds and are somewhat delocalized. This, however, inhibits rotation around the peptide bond itself, so that forms a pretty rigid structure. However, rotation is possible around the alpha carbon. This is important because it determines how a protein can form secondary structures, how it can form H bonds with other residues. So the polarity and torsion angles allow for different H bond formation depending on the rotation and residues. So the amino group acts as a hydrogen donor and the carbonyl group acts as a hydrogen acceptor. So talking about secondary structures, a amino acid chain can take on, for example, a alpha helix structure, which is a right-handed helix. H bonds form between every fourth residue, and it kind of loops around each other, with the average helix being 10 residues long. And another secondary structure is the beta sheet, so aligned strands of polypeptides that are oriented next to each other. They can be running anti-parallel, like right here, or in parallel, and they can be arranged from 2 to up to 12 strands next to each other. Usually they are connected by irregular structures, such as these loops right here. We need to have some sort of connecting structure between the sheets. And also, one protein polypeptide has more than one secondary structure, so usually it's a mixture between both alpha helix and beta sheet, depending on the residues of the amino acids and the amount of H bonds that can form. All right, this brings us to the three-dimensional structure of a protein, the tertiary structure. So besides the secondary structure, a protein or polypeptide can take on a structure in the three-dimensional space, which includes the backbone structure and the side chains. So for example, we can have these globular proteins, this little glob of protein right here, which, as we mentioned earlier, has a hydrophobic core, which makes sense. We don't want the nonpolar residues to interact with water, so they group on the inside, and this surface usually has polar or charged head groups, and is hydrophilic. Or we can have fibrous proteins, which are elongated structural organizations. So usually those are multiple helices that are wrapped around each other and form different sorts of filaments, and are usually important for structural features, versus globular proteins usually have functional properties. For the protein folding, usually that is governed by the hydrophobic effect, so the desire to avoid interaction with water. Again, we have hydrophobic residues that are folded on the inside of a protein, and then the hydrophilic residues on the surface. And usually folding is guided by that hydrophobic collapse, so the desire to stay away from water. And then once a protein is folded, it will maintain its stability through H bonds, ion pairs, or disulfide bonds. So summing this up, we have a amino acid chain. Hydrophobic collapse will then cause the initial formation of a secondary and tertiary structure, and hydrogen bonds and side chain positioning will then determine the tertiary structure in space and maintain shape and stability. Folding is usually not possible just by a protein by itself. Usually chaperones are needed to aid in protein folding. So even though the hydrophobic collapse drives protein folding, to reach the most stable and energetically favorable conformation, we usually need a chaperone. This can also help in proteins that are, for example, misfolded, or just took on a wrong shape, or, for example, if due to high heat, a protein got denatured and needs to be reshaped, a chaperone will come in and help reestablish the correct conformation. And then lastly, we have quaternary structures, in which multiple polypeptide chains can be linked together to form a quaternary structure. So if we have one polypeptide chain, and then we link multiple of those chains together, and if they're of the same nature, then we have a homogeneous protein. And if we have multiple different subunits, like for example, we have an A and B subunit right here, then those are hetero subunits. The number of chains can also vary in quaternary structures. We can have a dimer, which would be 2 chains, a trimer, which would be 3 chains, a tetramer, and so forth. All right. Touching on separating proteins really quick and how to manipulate them, for example. So if we want to separate a compound or a mixture based on the different properties, we would usually utilize chromatography, which separates molecules based on different properties, and usually is a mixture that is dissolved in a mobile phase, so a phase that carries the mixture, and it moves through a stationary phase, so usually a porous substance. Um, and based on the properties of the substances, the components travel at different speed through the column and then elute um, depending on those properties. One of those types of chromatography would be size exclusion chromatography. So we have the mobile phase that contains the protein mixture that runs through a stationary phase that, for example, contains pores of different size or beads that kind of catch those proteins based on different size and depending on the speed at which they run through this column, they elute based on their size. Another type of chromatography would be ion exchange chromatography. So again, the protein mixture is dissolved in the mobile phase, and for example, the column can be negatively or positively charged. Let's say it is positively charged. Clearly, all proteins that also carry a positive charge will want to get away from that as quickly as possible because like does not like. So it will elute first and run through the column really quickly. Next, we have uncharged proteins that really don't care about the charge of the column and they will just run through at their own speed. And then obviously negatively charged proteins will bind to the positively charged column and will run through the column at the slowest speed and elute last. So then, based on properties of the different compounds, we have now separated a solution. All right, next we're going to talk about protein function, and more importantly, oxygen-carrying proteins. So myoglobin is a oxygen-carrying protein that is important in our muscles. It is a globular protein, as you can see by this little glob right here, and is a single-chain protein. It contains a prosthetic heme group, so this heme group can carry out a specialized function, and the heme group contains a central iron atom or ion. So the heme group binds the iron with 4 nitrogen atoms and a histidine side chain that reaches into the prosthetic group, and a 6th binding component can be oxygen, which can reversibly bind and then change the conformation of myoglobin. So again, when oxygen binds to the heme group, it H bonds with another histidine group that also reaches into the prosthetic group chain and holds that a little down and changes the conformation of myoglobin. And this binding of oxygen is reversible. If we look at the binding curve as well as the affinity constant, so again, we have K here that we've seen before, which is the affinity constant, so the affinity that myoglobin has for oxygen, which is calculated by the free concentration of myoglobin and the free concentration of oxygen over the bound concentration of myoglobin to oxygen. And this will give us the affinity that myoglobin has to bind O2. So if we have a low K value, we have a really high affinity because this value will be really high, which will make the entire value low. If we look at the binding curve right here, we can see the saturation of myoglobin at different external oxygen pressures, so PO2, and on the y-axis we have the fractional saturation. And right here, this is the half saturation of myoglobin. So at about an external oxygen pressure of 0.25, we reach a half saturated myoglobin concentration. As you can see, myoglobin is pretty quickly saturated fully, so it has a pretty high affinity to oxygen. We can calculate the partial saturation of myoglobin with the affinity constant and the oxygen pressure. So everything is pretty straightforward. Next, we have hemoglobin, which is probably a bit more familiar to you guys. It is a heterotetrameric protein, so it has 4 subunits. It has 2 alpha globin chains right here and 2 beta globin chains right here. So heterotetramer because it is made up of different subunits. Each of the globin chains carries a heme group and therefore has that iron in the middle. It can reversibly bind 1 oxygen molecule per heme group. So overall, it can bind 4 oxygens. Also important is that myoglobin and hemoglobin are very similar in structure, but only share about 18% identical sequence. So that's pretty interesting that the globin subunit chains actually are pretty different in sequence, but share the same functionality and structure, which suggests a common evolutionary gene origin. All right. Looking at the saturation curve of hemoglobin, also compared to myoglobin again. So this is the myoglobin curve and this is the hemoglobin curve. We see that hemoglobin follows a sigmoidal binding pattern, which is due to its cooperative binding. So at low oxygen pressure, it actually has a really low affinity compared to myoglobin to oxygen. And it isn't until the first oxygen is bound to hemoglobin that the affinity rises pretty rapidly. So if you remember, the half saturation for myoglobin was around here versus for hemoglobin, it is at a much higher PO2. So overall, we can conclude that hemoglobin has a much lower affinity at equal O2 pressure than myoglobin, and as the pressure for the external oxygen increases, the affinity for oxygen increases as well. So talking about, the cooperative binding. So deoxyhemoglobin is in a tense state, and that is because the iron that is bound to the heme group is actually somewhat out of plane, so it is a little bit above the porphyrin ring. And once the first oxygen binds, it forms that H bond with one of the histidine residues, and that leads to the iron being pulled into the plane, which changes the overall conformation of the heme groups, which then affects the conformation of all the surrounding heme groups. So that allosteric binding of oxygen allows for conformational changes within the heterotetramer. So once we get over that first tense state, leading to a more relaxed state, so we see right now the binding affinity is really low for hemoglobin. It is in a tense state. We bind our first oxygen, and we see that the conformation of each of those subunits changes, and the more oxygen we bind, the higher the affinity for oxygen grows. This is actually really important because as we breathe in oxygen, we want to have a high affinity in order to bind oxygen that we're taking in. So we're breathing in oxygen, and we want to bind as much as we can in our lungs. So then the oxygen travels through our tissues and we can release oxygen to the tissues. So as we travel from lungs to tissues, the affinity for oxygen needs to change. High affinity in the lungs, low affinity in the muscles, so we can release oxygen in the muscles. So this actually is regulated by the Bohr effect. So the concentration of protons regulates oxygen binding and oxygen affinity of hemoglobin. So because we want to oxygenate our tissues, we need to have a decreased affinity in our tissues. And the more CO2 we produce from respiration, the higher the H plus concentration, so the lower the pH, and therefore we have decreased O2 affinity. As we travel back to the lungs, we have a higher pH and increased O2 affinity, so we are more likely to take in oxygen. Another substance that regulates oxygen affinity of hemoglobin is BPG, biphosphoglycerate. It is a compound that binds to the deoxyconformation, so the tense state of hemoglobin, and therefore decreases the affinity for oxygen. All right, and one last thing when it comes to hemoglobin and red blood cells. So I'm sure everyone has heard of sickle cell disease, which is a abnormally shaped blood cell. So it causes this sickle form right here. This is due to a mutation in the globin chain in our DNA. So we have a mutation from A to T. So if you think about the consequences of that, A to T leads to U for A in our mRNA, which leads to a different amino acid codon. So instead of glutamate, which is negatively charged, we now have valine, which is a nonpolar amino acid. The consequence of that is valine is nonpolar. It does not want to be on the surface. It wants to be away from the surface, hydrophobic, which then affects the structure of the protein. So we get this sickle cell. How is this related to malaria? Well, malaria is carried by mosquitoes and is transmitted through mosquito bites, which the parasite then infects our red blood cells. However, when the blood cells are sickled, so we have these sickled red blood cells, it makes it really difficult for the parasite to survive. So actually, if we carry one mutated gene for sickle cell anemia, sickle cell disease, we are resistant to malaria, which is a pretty interesting development, and which is why sickle cell disease is super common in areas where malaria is also super common. However, if an individual carries two mutated copies of the hemoglobin gene, they actually get pretty sick still, because it leads to this sickled cells blocking blood flow, so they aggregate and actually make it really difficult for blood to flow normally through our veins. All right, so we have talked about globular proteins, myoglobin, hemoglobin. Next, we're going to talk about structural proteins, so proteins that contribute to different structural functions. So we have actin, intermediate filaments, and microtubules. Those are all different types of structural proteins. So actin itself actually is a globular protein. However, via polymerization of these actin monomers, which is driven by ATP hydrolysis, multiple subunits can be added together and form this filament structure right here. The polymerization of actin starts pretty slow, actually, but once it starts, actin gets added rapidly at both ends, which is also called filament treadmilling and contributes to really dynamic structures within the cell. So it can rapidly and constantly change by adding and removing subunits at both ends. Next, we have microtubules, which are also made up of small globular protein structure subunits. And in comparison to actin, they are 3 times thicker and much more rigid than actin itself. So microtubules are formed via tubulin. So it is the subunit for the microtubules. There are 2 different monomers, alpha and beta, so it's also a heterodimer. So 2 tubulin monomers, alpha, beta, form this dimeric structure, and then multiple of those dimers come together to form a protofilament, and then multiple protofilaments eventually form this hollow tube, which is called a microtubule. Tubulin utilizes GTP instead of ATP as an energy source to polymerize, and microtubules are super important in cell division. So if you remember, in cell division, chromosomes get pulled to opposite poles, and this is done by these microtubules. So they need to be able to extend to the middle of the pole, attach to the chromosomes, and then contract to the opposite poles. So they also need to be able to change structure very rapidly. And then lastly, we have intermediate filaments, which are, as the name suggests, intermediate in thickness, so intermediate between actin and microtubules. One example is keratin. It is so the monomer of keratin is a single helix, and then 2 helices form a coiled coil, so they coil around each other. This kind of looks like a DNA structure. So these 2 helices wind around each other, but they can contain from 16 to 32 polypeptide chains. So 7 amino acid turns form 1 coil, and the first and fourth amino acid need to be nonpolar so they can interact with each other. And between the different monomer units that then link up to 32 polypeptide chains, they are held together via disulfide bonds. Another intermediate filament is collagen, which is the most abundant animal protein, and holds together and gives structure to the extracellular matrix. So a pretty important protein. It is also very unique in its structure in that up to 30% of the amino acids in collagen are glycine. This is because every 3rd residue in the helix, so collagen is also, the subunit is a helix, and every 3rd residue, which is in the center of the helix, must be glycine in order for the helix to form properly. Besides that, it is usually a repeating glycine, proline, and then a modified proline residue sequence, and then it arranges in a triple helix arrangement. And then multiple helices are then cross-linked together via modified residues. All right, enough about proteins. Next, we're going to talk about lipids and how different types of lipids are used in the cell. So lipids can be used for energy storage, membrane structure, and cell signaling, and can be grouped into fatty acids, triacylglycerols, glycerophospholipids, sphingolipids, and isoprenoids. All right. So the most common and simplest lipid is fatty acids. So this should also look pretty familiar looking back at exam 1. So a fatty acid is a carboxylic acid and has up to 12 to 24 carbons attached to it. Usually they are even numbered, So you will rarely ever see an odd-numbered fatty acid chain. Usually you count from the carboxylate end, count down towards the terminal methyl group, which is named omega. Fatty acids can be saturated or unsaturated. Saturated fatty acids do not contain double bonds and have the maximum number of attached hydrogen atoms. They are solid at room temperature because they are packed very, very tightly together. They don't have any kinks or bends in their chain. And due to that high packing, have a pretty high melting point. Unsaturated fatty acids have at least one double bond. They are usually in cis conformation, so on the same side of the double bond. If it was on the other side, it would be a trans double bond. Because of that kink or bend in the chain, they can't pack as tightly together, and they are usually fluid at room temperature, so they have a lower melting point. Humans cannot synthesize unsaturated fatty acids with double bonds past the number 9 carbon. As for naming of fatty acids, if we have an unsaturated fatty acid, we have the total number of carbons. So for example, if we have an 18 carbon long chain and we have one carbon, then it would be, sorry, we have one double bond, then it would be 18. One, if we have 0 double bonds, it would just be 18, 0. As for counting, since the last carbon is called omega, there are 2 different naming systems. Sorry. One is the delta system. So we start numbering the carbons at the carboxylate carbon and then count down. So depending on where we encounter the double bond, it would be counted from the carboxylate group. There's also an omega system of naming where we start counting at the last carbon of the chain. And then depending on where we encounter it, it would be omega -3, for example. Okay, our next type of lipid would be triacylglycerols. This should look pretty familiar because it is how we store energy. So what is found in our fat cells. It is a glycerol head right here that is linked via an ester bond to 3 fatty acid groups. Those fatty acid groups can be of different types, so they can be of different length or can be saturated and unsaturated, so they don't all need to be the same fatty acid group. The head can then be modified on the glycerol end, which can then lead to different functions of the lipid. So talking about head modifications, we have glycerol phospholipids. Again, we have a glycerol backbone, but we only have 2 fatty acids and a modification of the head group by a phosphate derivative. Glycerol phospholipids are amphiphalic because they carry that nonpolar tail with the fatty acid chains and a polar head group, the phosphate derivative. Next, we have sphingolipids and sphingomyelins. Sphingolipids contain sphingosine instead of glycerol. They only contain 1 fatty acid and have a polar head group. Those are usually components of animal or plant membranes. And lastly, we have sphingomyelins. They also have a sphingosine backbone instead of glycerol. Also only carry 1 fatty acid, but they always carry a phosphate head groups. Sphingomyelins are commonly found as insulating layers in neurons. I don't know if you maybe remember the name myelin sheet that wraps around and insulates neuron fibers. Our next category are glycolipids. Those are sphingolipids with 1 fatty acid, and they contain a sugar as a head group. So the structure should look pretty familiar to you guys too, if you look back at Unit 1 with the typical sugar modification. And then lastly, we have isoprenoids. as our last type of lipid. So those are lipids constructed from a 5-carbon unit that all contain the same carbon skeleton as isoprene. So it's a fused ring structure. It is a wide variety of biomolecules, so they can be of very different structures and shapes and carry out a variety of functions. So they can be classified into terpenes and steroids, and can be classified by the number of isoprenes, so 5C units that they contain. One of the derivatives of terpenes is a steroid. So we have that fused ring system right here, these 4 fused rings with this double bond that can be at any location throughout this ring system. And it can have very different substituents. So right here we have that OH, and here we have this carbon structure, which can be very different in different derivatives. Steroids are found in all eukaryotes and in some bacteria, and are important for membrane stability. Next, we have cholesterol, which is an amphipathic isoprenoid. It also has that fused ring structure right here, so similar to the steroid structure before. So cholesterol is a sort of steroid. It is a key membrane component, actually, and is a precursor for steroid hormones. I'll talk about the important for membrane structures in a second. So, as I already mentioned, amphipathic molecules can form a bilayer, with the nonpolar tails forming the hydrophobic middle part, that core center, and the polar head groups staying on the exterior portion of the bilayer. In living systems, bilayers are usually made up of glycerophospholipids and sphingolipids, and usually naturally occurring bilayers are a mix of different lipid types. So we don't just have one lipid type, but we have multiple different types. Usually, the bilayer is 3 to 4 nanometers thick, depending on the length of the fatty acid tails and if they are saturated or unsaturated. In addition, the size of the head group affects the packing ability. So the larger the head group, the less tightly packed the head groups can be, which then affects the fluidity of the membrane. So talking about membrane mobility, obviously, the longer the chain is, the less mobile the membrane will be. If we have unsaturated chains that have a kink or a bend, then they are actually more mobile because they cannot pack as tightly together. So more unsaturated components in a lipid bilayer leads to a more fluid membrane. Cholesterol comes into play here. Like I said, it affects membrane viscosity. So at low temperatures, it prevents super tight packing. So the lower temperatures are, the more tightly the lipids want to pack together, but cholesterol kind of sits in between them and avoids that tight packing. And at high temperatures, it does the opposite. They want to be really fluid and far apart from each other. But with cholesterol sitting in between, it prevents like large gaps forming in the bilayer. So it keeps them kind of together and acts as a membrane buffer. Okay, in a membrane, different regions are characterized by different fluidity levels, and the bilayer is made up of very different components. You've probably heard the term fluid mosaic model. So we have a very fluid membrane that can move around. We have different components of the bilayer. We have proteins, we have lipid rafts, we have sugar modifications with different functions. We can have transverse diffusion from one leaflet to another, so they can flip-flop around the membrane. We have lateral diffusion within one layer. So overall, there is a ton of movement within the bilayer. All right, talking about membrane proteins, we have three different types. The first one would be an integral membrane protein, which spans the entire bilayer. It's usually some sort of helix that has a lot of hydrophobic residues, so it can span on the interior, whereas the hydrophilic parts of the protein sit on the outside. Next, we have peripheral proteins, which sit on the outside of the bilayer and are usually free to move around. And then we have lipid link proteins, which are anchored into the membrane via different modifications. So we have myrosylation, which is an irreversible lipid link. We have palmitoylation, which is a reversible link, and then we have prenylation. So different lipids can link a protein into the lipid bilayer. All right, as I already mentioned earlier, the plasma membrane is a barrier to free diffusion, so things cannot just freely pass through the bilayer, especially because of its very hydrophobic core. So we have different concentrations of ions and other substances across the bilayer. Therefore, we also need those membrane proteins that we just talked about to carry out transport through the bilayer. But more importantly, we need to be able to transport ion across the membrane to keep the membrane potential, which I explain in a second. Yeah, so we have a membrane potential, which can be calculated through the temperature and the natural log of the ion concentration on the inside versus on the outside. So talking about the membrane potential, again, it is caused by different ion concentrations across the membrane. So we have a higher potassium concentration inside the cell and a higher sodium concentration on the outside of the cell. We also have other ions, usually negatively charged ions, on the inside of the cell. So overall, that causes a resting membrane potential of -70 microvolt. So the inside of the cell has a slight negative charge, and that is the membrane potential. In order for cells, for example, to communicate with each other, it is important that we can change that membrane potential. So action potentials, for example, cause muscle movements. This kind of looks like a heartbeat, which is essentially what a heartbeat is. So that kind of electric current. So that is caused by action potential, which is a depolarization of that resting membrane potential. So depolarization means that the potential gets less negative, more positive. So from that -70, we depolarize the inside of the cell to about -40, which is essentially the peak of an action potential. And then the cell will repolarize and we go back to the -70 microvolt. This depolarization happens through the influx of positively charged ions into the cell. So in our case, we get sodium ions that flow into the cell, and then in order for repolarization to achieve the resting membrane potential again, we need to remove positive ions from the inside. So we remove potassium ions from the inside. And the resting membrane potential is kept up by the sodium-potassium pump, which I will talk about in a second. So we already know that we cannot have free movement across the membrane usually, but there are some ions or small molecules, actually not ions, small molecules that can cross the membrane by itself. So usually those are nonpolar molecules. They can just freely diffuse across the membrane. Others need carrier proteins or channels, and those are needed to, for example, transport ions or larger proteins. So simple diffusion does not need any carrier proteins or channels. Facilitated diffusion does need carrier proteins or channels. Passive transport, also important, happens following the concentration gradient from high concentration to low concentration to establish an equilibrium. So talking about facilitated diffusion, we have those porins, which span the entire membrane, and substances can pass freely through those porins. So it's essentially just an open channel. It is always open and usually transports ion and other small molecules that cannot pass freely through the bilayer with their concentration gradient. Next, we have aquaporins, which exclusively transport water. So those are very specialized channels only for the transport of water. And then lastly, we have Gata channels. Those also span the entire membrane, but they are open or closed depending on different circumstances. So we have voltage gated channels, which open based on the voltage. So if you think about the membrane potential, if we want to transport potassium or sodium ions in or out of the cell, we need to have a gated channel. As the action potential gets depolarized, we want more sodium ions to flow in. So the gated channel will then recognize that change in membrane potential, and the sodium channels will open for more sodium to flow into the cell. There are also mechanosensitive channels that respond to membrane tension and open or close depending on a change in tension of the membrane. So conformational changes in those channels happen due to external factors. They bind molecules or open to molecules diffusing through the membrane depending on external factors. And lastly, we have active transport, which requires energy input, usually flows against the concentration gradient. So passive transport flew with the concentration gradient. This is against the gradient, which is why it requires energy input. So again, back to the sodium-potassium channel, we need to move the sodium ions against its concentration gradient from inside of the cell back outside of the cell. This is done with the help of ATP. So 3 sodium ions bind from the inside to the channel. It also binds ATP, which is then hydrolyzed. That energy is then used for a conformational change from this position 1 to this position 2. Sodium is released to the outside, and potassium, 2 potassium ions can bind on the channel. The remaining phosphate from the hydrolysis of ATP is then released for a conformational change. So the energy from that phosphate release is used for a conformational change back to the original conformation, and potassium is released to the inside of the cell. So this antiporter, I forgot to talk about this on this slide, transports 2 potassium into the cell and 3 sodium outside of the cell. Jumping back to this really quickly, so we're talking about an antiport where one substance gets transported in one direction and the other in the other direction. A symport would be if they get transported into the same direction. And this also brings us to secondary active transport, where movement against a concentration gradient is coupled to the passive transport of a secondary substance. So looking back at the symport, for example, if this red substance is transported with the concentration gradient, it does not require energy input. This green substance, however, is transported against its concentration gradient, but because it can be coupled to this transport right here, it does not require external energy input, and it just can kind of use the same channel as this one. This can also happen in the other direction, for example. All right, I hope this covered all of it. I know this was probably not a comprehensive review, but I tried to cover as much as I could, and I hope it was helpful. If you have any questions over the weekend, feel free to reach out, to email me. We can set up a meeting before, or we can just meet over Zoom. So just let me know if you have any questions or if you need anything. Good luck studying, and I hope this helped.